without further ado, I'd like to introduce the moderator of tonight's program, Luka Mircovic. Luka was born and raised in Belgrade, Serbia, and emigrated to LaPorte, Indiana in 2007. Luka graduated from Northwestern University in 2012 with a bachelor's degree in communications. While at Northwestern, Luka, who stands 6'10" or 11?
I think it's like 11.
Oh, okay, all right. Sorry, I don't want to cheat you of an inch. 6'11". He was a power forward for the Northwestern Wildcats basketball team from 2008 to 2012, where he was noted for his gameplay intensity and connection to Wildcat fans. Luka also obtained his MBA from Chicago's North Park University in 2015. Currently, he is Director of Operations at Adams Third Wave, a Chicago-based progressive national goods transportation company. Luka has been a Chicago resident since 2008. Tonight's featured speaker is Neil Kelleher. Neil is a distinguished molecular bioscientist and chemist at Northwestern University. He runs Kelleher Research Group that invents new ways to understand how human cells work at the molecular level. He also heads Northwestern Proteomics, the leading lab in the world for top-down proteomics approach to measuring proteins. Neil is the founding president of a worldwide research consortium aimed at mapping 1 billion proteins throughout the human body. The result will lead to new discoveries in human health, including new antibiotics and anti-cancer molecules. His record of achievements and impact in the field of proteomics is long, and his research is bringing us closer to a cure for cancer and other diseases. So, ladies and gentlemen, I bring you Luka Mircovic and Neil Kelleher.
Thank you all. Thanks for coming out tonight, and I wanted to really thank Luka for joining me and bringing in a great group together to talk about some science. And science enthusiasts and lay public, all questions are entertained and entertaining. I wanted to thank C2ST for hosting tonight and our event, The Long and the Short of Proteomics. We'll talk about human height, and that's a surrogate into human disease research. And so bringing it out into the streets of Chicago, the Emporium, I want to thank them as well. And thank you all for coming out. Let's have some fun in the next hour or so. Yeah, so one of the things we talk about in height is where does it come from, right? And how do you understand that? And that's something that Luke and I talk about and joke about, but we wanted to bring that to you in a slightly more serious tone and think about lots of different traits for human beings. And so when you think about two human beings, you might say, what's the same and what's different, right? And there's lots of traits that you could think about, like we're male, and if you could think about others, you might think about similarities and differences. We both walk on two legs most of the time.
Yes, yes.
Although I've been called worse, so
Yes, for sure. Same here.
Sometimes I scramble. I'm low to the ground, so that's how I roll. We're both carbon-based life forms. We have symmetry. Humans have a body plan that include bilateral symmetry.
So we're both really good looking? Is that what it is?
Symmetry does connect to looks, but
Okay.
I would say I'm way better looking.
Okay,
Like,
I don't know about that, but
I mean,
okay.
objectively, we could take a poll.
Sure, I mean, that's
Okay.
fine. We can talk later.
We'll table that.
Okay, okay.
Opposable thumb, so we can grab stuff, right? And a long lifespan. We both will live to 72.
Although you're way older than I am.
Yeah, that's
Just
true.
to clarify.
Oh, that one hurts.
Okay, just to clarify.
I'm 49, but I did lie about my age earlier tonight.
Okay.
So if I'm honest, yeah.
Yeah,
But,
okay.
Yeah. Yeah, and so when you think about the genome, the human genome, we have largely the same DNA. We really do. Except for, 10, perhaps at most 20 million little spelling differences among the 3 billion letters that are in our human genome.
Mm-hmm.
And so, I must have a lot more of the pretty 10 million letters.
Oh, is that what it is?
I think so.
Okay, all right.
Okay.
All right, well.
But somehow you're much taller. And I think generally people think that's more attractive. Like,
right.
You know,
I have the accent too going.
Oh.
You know, it's
Oh.
kind of.
Right, tall, dark, and handsome.
That's right.
You don't hear short, dark, and handsome very much, but I'm trying to bring it into a thing. And there is one major human trait that differs between us two, and
That's hair, right? Or no?
yeah, that's
I
right.
think it's hair.
That is a beautiful thing. I don't know. Some people
Because
say
typically, like usually when you and I hang out, like people will be like, "Are you guys brothers? Are you guys related?" We get that a lot. Right?
Yeah.
I mean-
well, bald is beautiful. I gotta say. I
oh,
gotta
for
give it
sure.
to you. Right.
Yeah.
But just for contrast, there is one human trait
Okay.
that's quite obvious. So let's just take a moment
Okay.
and stand up.
Okay, sure.
So to just
Sure.
see the variation.
Okay.
And we're of course not the first to notice this, right? I mean, if you think about the variation of human height, etc. Here's a picture of Willie Shoemaker and Wilt Chamberlain from some years ago, and they too have very extreme traits when it comes to height. So yeah, and when you think about this natural variation and what controls human biology, things get complicated pretty fast. But in the next, half hour, 45 minutes, we're gonna wrestle this thing to the ground. So how do you feel about that, Luke?
No, I'm excited. I'm excited for sure.
Yeah. So he's on one end of that curve. As you look on the right side of this slide, he's on one extreme, and I'm on the other. But we can still be friends, I think.
We'll see. It's debatable. Debatable. Yeah.
Depends how aggressive I get.
Yeah, I mean,
right.
I'm only here for the money. They're paying me big bucks,
I know.
top dollar. So,
Right.
I mean, I'll let
Well,
you
a talent like you, I mean, I could dunk the basketball.
Oh, really?
Pretty in my
Wait,
day.
For like your kids' hoop or?
No, I had to like. There's actually a Twitter video of me dunking the basketball, and it is exactly on a 7-foot hoop.
Okay, that's pretty good actually.
I mean,
all
you-
things considered, that's
Yeah,
That's
but there's
pretty good.
nothing else in the shot, so you can't really tell
Right.
that it's 7 feet tall.
Right,
And,
for sure.
it went-
No, I'd say this. Maybe this weekend you and I can play one-on-one.
Really?
And then we can document that.
For money?
Sure.
Yeah,
for
I got this
anything.
move where I go
For-
through his legs. I can dribble the ball
Okay.
through his legs and like,
Okay, sure.
come up on the other side. And what's there? It's the hoop.
There you
on
go.
the
It's
other
right
side.
there.
That's right.
Yeah. No elbows though.
No.
No elbows, please.
I have noticed that when I play basketball, I catch elbows in the teeth, and then
Yes.
I go to the dentist.
For me, it's the stomach.
I aim a
just-
little.
yeah.
I aim a little lower sometimes.
Yeah.
But yeah.
Okay.
Right.
All right.
But one's more expensive than the other.
Right.
So, there are other examples of phenotypic variation or traits. The phenotype is a funny word that biologists use to talk about a trait. And this trait of height is often described. Here's another even more extreme example of two basketball players. So I first met Luca some years ago, and I said I love to play basketball. And at 5 feet tall, I think speed will help me.
So it was like Muggsy Bogues, like your childhood hero.
Totally.
Loved him.
He was totally
Loved him.
my hero. I'm like Muggsy Bogues. When I was young,
He was the. Which
long-
was a long
time ago.
Was that like the Eisenhower administration? Or was
yeah,
it
that's right.
No?
That's,
Okay.
that's about right. Yeah, got a gray hair here.
Okay.
Yeah.
Here's my thing. Is like, I mean, how does DNA define how tall we are? Like, is it only DNA, or is it anything else like nutrition or like pollution or things like that, or exercise? Can you talk about that a little bit?
Yeah, so your DNA, it's sort of like your blueprint, right? And it has a strong component. And in human biology, there's a strong component of your height that is determined by genetics and your DNA and your family, et cetera. And the NIH estimates that about 80% of that is determined somewhere in your genome, somewhere in your DNA, which contains information for about 20,000 human genes. That's what's encoded in your 3 billion letters of your genome that's throughout most of the cells in your body. So it's a lot. 80, the family, you can tell your father was not my father.
Yes, correct.
How tall was your father?
My dad's probably about 6'7".
Yeah.
And my mom's probably like 5'10", 5'11".
So both pretty tall.
Yes.
On the distribution,
Yes.
if we go back, yeah.
Right.
This is the men's distribution of height, and
Yep.
but yeah.
How tall were your folks?
My dad was 5'3",
Okay.
is 5'3". Hi,
Okay.
Dad.
Okay.
Is, but he's
Okay.
still living.
Okay.
And my mom is 5'4".
So that's tough to, like, how did you manage to beat them with like,
Well-
you're 5'3" and 5'4", you would think that like you're going to be a little bit taller than
You
them.
would think that.
Right?
So why am I
So
5 feet tall?
how does that work? I would
I've
think-
spent my life asking that question.
Yeah.
And so the little portion of this pie chart that's in blue is what you were asking about. It's sort of nutrition, environment, not just the playbook of your body and your DNA and your genome. And so in this estimate, about 3/4 of your information is spread around these 20,000 human genes that define height. And so there's many genes that contribute into that trait. And so I actually, when I was 16, 17 years old, I was a wrestler, and I was trying to make my coach happy, and he needed a wrestler at the 101-pound weight class. And I was 117 pounds when I entered practice.
So you had to cut weight and all that
I
stuff?
cut weight down to 4% body fat as I was a young person, and I think that's my idea of
I mean, honestly, I wouldn't be surprised because a lot of wrestlers are kind of short. Let's be honest,
It's
right?
interesting,
So
short.
probably cutting weight, right? So that doesn't help.
Yeah.
No, I mean, so just theoretically, like if you were predetermined by your DNA to be 6'5", but then let's say due to poor nutrition,
Sure.
like malnutrition,
Young in life.
dehydration,
Yeah.
But again, is it mostly like in early life, like, you know, like I
Yeah,
don't know.
well, it's the growth, the
Yeah.
growth plates in your bones, and your bones grow, and you have to have cartilage, and this is proteins. Your bones are you're proteins, right? DNA is the body plan, but the proteins build you. And somewhere in there, they started to not really grow, and nutrition is definitely part of that. You know, if you're starving, you're going to be smaller. But it is that blue part of this pie chart that is the minority, at least for this trait of height.
Gotcha. So wait, so does that mean that like, if my parents were giving me more food, I would have been like minute bowl 7'7"?
It's a thing,
that-
right? I
right?
mean, no, but there are extreme diseases that, not that you have a disease.
Okay.
I'm not saying that.
Thank you for clarifying.
I'm saying like growth hormone can be overexpressed naturally with some tumors are known to do this. Some other very extreme mutations in the human population, gigantism, right, or dwarfism. These are genetically caused in large part. But yes, your nutrition would alter your body plan to some degree. You still would have been very tall.
Sure.
But you would have been shorter or slightly larger had you optimized.
What about like pollution, air, clean air, stuff like that?
I think anything toxic that affects your DNA and it may not mutate your DNA, but can very much affect you. Yeah, if you're living in a seriously toxic environment, malnutrition, you're going to be smaller. You're not going to thrive.
Okay, that makes sense actually. Okay.
Yeah, and so it's the conversion of the blueprint. And this same article about the DNA, that there's an estimate out there that like 700 different genes in the genome affect height in some way. So it's this really complicated interaction between your genome to a trait, your height in this case. And as we'll get quickly to some other traits like cancer, disease, and other just natural variation that we call health and wellness.
Okay. All right.
Yeah.
Good stuff.
So if you have questions, Chris will bring around the mic and you can interject any time. A two-way street is totally welcomed here. Yeah. So getting to that point about traits, height being one of them, a fun one to talk about that's very striking when you see two people at very different ends of the spectrum. You've noticed a lot of different traits about people you love and your family members, people out on the street, right? And there's a big variation on a very small number of changes in the human genome. You're talking about 10 million letter changes among different humans. We're very actually inbred as a group of 8 billion people. But if you think about now disease, this is when a family member is stricken with one of these diseases on the right of this slide. Those two are traits. And the main theme of tonight is to move from genes and the human genome, which we now understand at base level, to how do these traits get formed, how do they get affected, and then what can we do about them when we don't like them, like disease? How do we intervene? Why is drug development relatively inefficient? Why can we not have deterministic operations with human biology? And it's in part due to the fact that we have not regularized the world of human proteins like we did the genome 20 years ago. The proteins, we're built of proteins. That's what's underlying all these different traits, is proteins. So like your hair, your bone, etc.
I know you're going to talk about the Human Proteome Project a little bit later, but can you give us a timeline as far as like how close are we to identifying all of it?
Yeah, so human proteins, we know we have 20,000 genes, right? But in different tissues of your body, like your liver, your brain, the same gene can produce different proteins. And that's what the theme of tonight is about, is trying to regularize just what is produced in health and wellness so that we can detect disease early. And that's the theme of this evening. And I would say that in something like a dozen years, we can at least set the table properly, that we can determine all the different kinds of proteins that are produced from our genes. So about a dozen years.
Gotcha. Okay. Sounds good.
And the scale of the genome, it's something on the order of the scale of the Human Genome Project.
So it's still pretty early. I mean, would you say it's kind of like in the 90s where it was like with the Human Genome Project?
Yeah.
We're at about a similar timeline right now.
Yeah, no, that's right.
Yeah.
I mean, so in like 1989, there was a group of like 25 geeks. They went up to Alta, Utah, like the ski resort, and they said, well, could we actually think about sequencing the entire human genome? And we knew it was about 3.2 billion letters, A, G, C, T, right? And in 1989, you didn't know if this group had been drinking too much or that they were really visionary and onto something. And yeah, I would say it's about similar. By the year 2000, 2002, the Human Genome Project was done. There's President Clinton shaking the hand of folks at year 2000. It was an amazing thing that happened. You brought physicists, biologists, computer scientists, technicians, technologists, engineers. They all swarmed the problem, and we sequenced the human genome so that now, if I had cancer now, I would have the cancer genome sequenced. Because of all of that investment in technology, we brought hundreds of thousands of new jobs into the private sector. I mean, it was an amazing project. And so inspired by that, I've taught that in my classes. I'm among a growing chorus of voices saying, hey, let's run that script again
and
for proteins.
Can you tell us about, like, who are some of the other folks that are involved with this?
Yeah, so there's a consortium for top-down proteomics. That's a group I've formed. It's like me and 3 friends in a basement. No, there's like 450 scientists from around the world that have joined in that consortium to begin to say something like that we need a centralized big science project focused on human proteins.
But Neil, you mentioned to me before, before when we were prepping for the presentation, you mentioned that also the general public, the role of the general public in the Human Proteome Project is really important. Like, and that's something you've been trying to push too, right? It's to try to get younger people involved and just pretty much getting everybody on board, right?
Right, and I think the Human Genome Project, it costs something on the order of $5 billion.
I'll cut you that check after.
Yeah, do you?
I got you,
right.
Man. Don't worry. Don't worry, man.
Yeah, so that's a B with billion. That's 5. And there was this moment where they said, all of the 3 billion letters that we're not going to tee up this project until it costs $1, $1 American dollar per base. And that meant it was a $3 billion project. And guess who doesn't fund stuff like that? Companies. Guess who does fund that? The government. World governments, in this case the Genome Project was the American government, so you all, taxpayers, and so the public's opinion absolutely matters about this. And it's partly why we come out here today to just socialize it. Your questions, what's not clear, what would be the benefit, etc. Those really, there's a question there. They help me to think, okay, justify such a project, right? Because there's a lot of things we could spend money on. You have a question?
Hey, hi, thanks so much. Has this Human Genome Project, all of the human genes are in the public domain, right? None of it has been privatized, correct?
That's right. The U.S. Patent and Trade Office, USPTO, actually said you cannot patent a gene. Because people who first found the breast cancer gene, the BRCA1, BRCA2 genes, people tried to patent that and monetize it. And the USPTO said, and the Supreme Court actually in a famous decision said no, you cannot patent natural genes. So you're right. Yeah. And there was another question up here. Here we go.
The first genome, whose genome is that? The one publicly? We have a thing from Science magazine, after the first genome was done, but it doesn't say whose genome is that.
Right. So you may remember there was a public and a private effort. There was a publicly funded effort that was 22 human beings, and the private effort, like here came this company, and that was Celera Genomics, and that was 5 people, one of which was Craig Ventnor himself. And so it was 27 people's genomes, and that's an important point because people sequence those genomes just of healthy people. But when it comes to justify it to the American public or the worldwide public, why would you do such a project? Everybody wants to talk about disease. It's going to improve our condition to detect disease, fix disease when we get them, etc. But the Human Genome Project, it's like first we had to get the technology right. And that's what the Human Genome Project did. That's what that $5 billion did. And then in the decades after that, then the companies started to get all these new jobs, and then it became now ridiculously cheap to sequence the human genome, about $1,000. There's one way in the back there. These are great. The Q&A is started already.
We start early.
Right on. One up front and then one here. Then we'll get back on track.
So for the Human Genome Project, you were able to work on, you said collectively 27 people.
Yeah.
But for proteomics, you stated that you have different proteomic kind of characteristics for even different organ systems. So isn't it a much more,
Yeah.
right? Isn't it a much larger undertaking than the Human Genome Project because you're looking at microenvironments and you're looking at proteins within different ages? So would you consider it a different type of question and one that wouldn't really be completed as the human genome was, because you would constantly be discovering new environments?
That's a beautifully framed question. So what you just said describes why scientists can't really agree on what a project would be. What would a human proteome look like? Oh, you want to do 60-year-old people, 20-year-old people, a mix? What? And liver tissue, cells, or brain neurons, kidney cells, right? Because they're very different. Your kidney's different than the liver, even though it emerges from the same genome. So it makes it a much harder challenge to get scientists to agree, much less funding agencies. And so I have joined with some people to try and frame what a project would be. And that frame is based on cells. So here we have like these Russian dolls. You know, we're built in different layers. So my body has within it tissues like the liver and which we can challenge this evening at the bar to be functional. And then within
I won't tell your wife. Just to
Okay,
clarify.
that'd be great.
I got you.
It'd be great.
I got you. Don't
Well,
worry.
if I do drink a little too much, you're going to carry me home.
Sure.
That's the thing.
I'm pretty sure there's a photo somewhere of me like giving you like a big back ride, like, you know.
I do
doing your
remember
bag.
that.
I'm pretty sure there's one.
I do.
But we will not be sharing that one with the general
Okay, so
public.
sidebar. So I'll come to the question, but sidebar. So when I play a lot of basketball, I love to play basketball, and when I see very tall men, at least two of them walking together, very tall people, I will jump and push off their shoulders and like do this sky thing. I used to jump and I tried that with him. I could not jump high enough to reach, so I'm like climbing up him one day on the street. Yeah, that's what happened.
That's what happened.
Anyway.
But by the way, Neil, just before you get into the
That was a great
answering this
question, yeah.
wonderful question, can you just give us just a high level overview as far as like what is proteomics and then sort of, you know.
Yeah,
talk
sure.
about the
Let's
rest,
we'll come back to that question. But the quick answer is it's a billion measurements. It's a billion different proteins across the human body. That's one answer to your question. And to be able to measure those for $1 each. So it's inspired by the genome model, but the analogies break down pretty quickly. So, but yeah, you're right. Let's give a chance for.
And I apologize.
Yeah.
I started asking too many questions, and then
No,
so.
There's no right way to tell this story. It's so for the layers that were built up, you got these Russian dolls behind us, just showing that we built up in layers. Like within my liver, as a tissue or organ, there are these cell types. There's different cells in there, or in the blood. You have almost 300 different types of cells swimming through your body in the blood, right? So within cells, what do you have? What makes up cells? What makes up us? What makes up traits? The answer is proteins. Proteins, hair, eyeballs, etc. So they are linked to all kinds of human disease. Every human disease, its progression, its extent, whether it can be reversed, it has to do with proteins. And so it seems like creating the definitive reference list of what proteins are produced is fundamental to understanding human biology. So that's what proteins are. They reside inside cells mostly. Some are free and in your cerebral spinal fluid or in your plasma or serum of your blood. There are proteins swimming around that are not in cells, but many of them are in cells. And cells are like a city. They're just like Chicago. There's this really complicated trafficking system, communication system. I mean, a single cell is an incredibly complicated thing. So when you think about proteins, you know, they have 20,000 human genes, and they create all these different proteins that occur in different flavors. So from the single human gene, you can create dozens or hundreds of different proteins from the same gene. And that's what makes a liver cell different from a kidney cell or a neuron, et cetera. So that's proteins. And proteomics, that's what is the study of proteins. And you know, they matter in all sorts of disease phenotypes.
So how do proteins affect us individually?
Yeah, so in all sorts of intrinsic ways.
Mm-hmm.
I mean, when you think about, okay, I have a tumor growing on my kidney, that is cells proliferating too much, right? When you were young and growing, your cells had to proliferate and make more of them. That same sort of biology has now got errors in it, and it's run wild, and it's generated a tumor, and that's cells and proteins going wild. And we are desperate as a society to try and put the halt on that. And as we try and do that through toxic drugs or protein-based drugs, proteins are drugs, and that's what's on this slide, about why proteins matter. That they're the biggest types of new drugs coming out of pharmaceutical companies are proteins themselves. So we're trying to fight proteins with proteins. And this fight, we are winning slightly improved rates, but how do we really win, right? We have to understand all the proteins. Like, because cancer cell proteins are different than normal proteins, clearly. The cells are different, and cells are built up of proteins. So that's why proteins matter. And the proteomics is simply, that's a funny word that just means the study of all the proteins. That's the field of proteomics, and that's what I'm in, and that's what we do at Northwestern Proteomics.
Awesome.
Yeah. Yeah, please. And
what is the earliest time that you could diagnose these proteins and see inside the uterus, the fetus, can you detect these proteins and see this fetus has these chances of cancer, or how tall this baby is going to be? At what age you could
Right.
detect these proteins?
yeah, so when a fetus is made and is growing, it's growing proteins. It's reproducing cells at a very fast rate. Cells are growing, the sperm and the egg get together and you get 2 cells and then 4 and then 8 and then, in very short order. And that growth is driven by proteins because you're growing an organism, you're growing a human. That's the bulk of that mass is proteins. The DNA is a small amount of the mass. But to answer your question, the detection of disease is probably best left to DNA. You can actually detect the DNA circulating in the blood of the mother. You can detect fetal DNA circulating in the mother and actually diagnose without amniocentesis. This is reality today. Why is it a reality? Because we invested in the Human Genome Project 20 years ago. That's why it's a reality. So I wouldn't be sampling a fetus to look at its proteins until after we had an incredible regularization of the human proteome, like we did the genome. So that's the answer today, but in 20 years, could we be analyzing proteins with incredible specificity, sensitivity? Yes. Another question there.
So who's going to be funding this, and are the proteins going to be in the public domain and not in the private?
Hopefully, U.S. government would be one of many governments that would kick in for such a project. And yes, totally public domain. If the public is supportive enough to get governments behind it, because I don't see companies, they're not going to line up. That's the lesson of the Genome Project. To do something this innovative and requiring a technological leap, it's the role of government. And or philanthropy. Some of the early grants that we've been able to get aligned with this project, like for example, Paul Allen, the Paul G. Allen Family Foundation, has funded a small pilot toward this project. And so philanthropy, and that's another role to try and catalyze it. But at scale, I think it has to be the role of governments. And that means it's in the public domain. Violently, yes, in the public domain. Yes. I don't know if we got to your question that was so beautifully phrased earlier about how do you frame the project. Maybe let's get through 1 or 2 more slides and get to the very end, and that's the crescendo is the answer to your question. Yeah. Any other things you're confused about, Luca?
No, I think I'm okay so far.
Yeah, you got the stock market figured out?
Yes, I got it all. No, I'm telling you, yeah.
That's excellent. I have this analogy to help people understand proteins a little bit better. Mm-hmm. I liken it to like the coach's playbook.
Okay, finally we're finally.
Yeah.
You're like speaking my language.
Yep.
I finally understand.
Yeah. Well, so
Yeah.
when you played for the Wildcats,
Okay.
you had lots of set plays, right?
Yes. Hundreds.
Hundreds. Yeah.
Yes.
Right. So that's the playbook, right?
Okay.
That's the coach is drawing it up.
Okay.
Right. So the analogy in this metaphor, it's that's the DNA, right? And okay, can you, Luca, or can you not predict the outcome of a basketball play?
No.
You have to run the play.
I have to run the play to see if it's going to work, right?
And that's life, right? Can you take your genome of a young child and predict what's going to happen? Lord, no, you cannot. You can't predict the height with precision. You can predict something general about it, But that's even hard because there's 700 genes involved, all contributing in ways through proteins to grow a human body. We don't understand that. So, for understanding disease, if you know that is the plays of life that you run every day, right? And so, that's why you got to run the plays. And the proteins experience, they absorb problems, they help fix. All of the proteins in your body right now, they are your worker bees. If you're going to go out and smoke a cigarette, you know who's going to repair that damage? Proteins. That's right. Proteins come in and will repair that damage. So that's life. You're alive, and that's what...
But Neil, you're saying through the Human Proteome Project, we will be able to do a better job of predicting some of those things?
Yes. It will take a long time. I'm careful not to overpromise, but it's a key part of understanding the parts list of what we're built of. Mm-hmm. And then we have a lot of range of variation. So how are we going to predict or detect your cancer risk or your progression if you're at stage 2 or 3 cancer with precision, right? Not some vague idea, but before it becomes something you can image as a tumor. How to detect it early, monitor you if you have certain risk factors. This is part of, and then really the interventions. Or if I'm going to create a new organ, like I need a new heart. I will need a new heart.
Oh, really?
Yeah, in like 2050.
Okay, 2050.
Totally planning on it.
But you're already pretty skinny. I feel like you take care of yourself.
I'll go long.
You don't need that stuff, no?
I'm working on it.
Okay. I did see you eat Mexican food before here.
I did overeat a little bit. But if you're going to create a new heart, you really, for my body, you have to understand the cells and the proteins within them. If you want full bottom-up control over building the human heart, one that's going to go in my body and actually give me extended quality life, you've really got to understand the proteins because it's built of cells and proteins. There's a question up front. Yeah.
Is it not against science to be precisely predictable for each person? Like a pseudoscience, astrology only predicts precisely, but real science gives epidemiological prediction, but not a specific to a person. Because you cannot say this person will get when he's 62 and 0.235 age, he will develop cancer of the lung. You cannot say that because that's against second law of thermodynamics.
You're exactly right. You can't predict that if I'm 39, as I am now.
Yes, correct.
I'm 39.
Yes. You and I went to college together. You and I graduated.
Right, remember that time?
Yeah, man. We went to the same fraternity, all that stuff.
I'm actually 49. But you're right. If I'm 39 and you say you have, you will develop a detectable stage 3B cancer when you're 62. That is not, but what you can do is say you have a higher than normal risk, and that's where DNA and proteins come together to, you need to watch your, mutation load because you have some oncogenes, genes involved in cancer, that are already mutated. You're hanging by a thread, so you really need to watch it. So you monitor carefully the proteins, and and images also taken by medical imaging. Proteome. You're right. Proteomics will not be the be-all and end-all of precision medicine, but right now it is a really open, undomesticated territory. It's a wild frontier where the detection of disease based on proteins is not reliable for many, many phenotypes. I can give you one example, like prostate-specific antigen. It's a protein, PSA. Maybe you've heard of the PSA test for men. I think in a few years I'll be getting such tests. And it's not a great test, right? Why is it not a great test? That's because the PSA, the protein, it's not just one protein. It occurs in like 100 different variations, some of which are elevated in cancer. And that's the kind of thing that my lab is detecting and then trying to layer on with precision those 100 different proteins. Right now, our assays lump them into one, and it's just imprecise. So hopefully that answers your question the world has many ways it can measure things. But I guess in the last few minutes, maybe we can come to the crescendo point and answer that great question. How would we attack the human proteome and try to measure all these different forms of proteins, like the 100 different forms of prostate-specific antigen, the protein? One answer to this, and an answer that I get 400 of my close personal friends in the consortium that we formed, it's a worldwide consortium, is to base the project on cells and cell types. And so in that liver I talked about that had lots of different cell types, the idea of this project would be in each type of those cells, or in the 300 different cell types in your blood, that within each type of cell to determine 250,000 proteins and to measure them with precision, and that if you estimate that there's 4,000 human cell types, which is a decent estimate, you multiply those two numbers together and you get to 1 billion measurements. So there's 4,000 cell types as an estimate, 250,000 in each cell type. That's 1 billion, what we now call proteoforms. Those are the 100 different forms of the PSA. Those are 100 proteoforms. And the consortium that we founded, that's a word, proteoforms. We view it as a movement. It's not a word, it's a movement, the proteoform movement. And that by measuring those with precision, 1 billion measurements across all the different cell types in the human body, we will regularize the human proteome. So that's the long-winded way to answer your great question there from the back.
Awesome. At this point, I think we should maybe go back to the questions again, if anybody has any further questions. I think we got some in the back, Chris.
Yeah, awesome. We got like 5 of them.
All right.
Throw darts, fire them. Yeah.
Also, guys, I can answer any questions related to basketball. Why didn't the Bulls get the #1 pick this year?
Oh, that was, they only had a 12% probability.
12%, but yeah, so.
So you have 400 different, or 400 plus different colleagues around the world, and you're going to be doing this. How are you going to be organizing this? Is it going to be, Illinois gets liver cells, of a certain variety, and someplace else gets a different type of cell?
Yeah, so that's a great, organizationally, the way the Genome Project went was there were 5 major centers that as the engineering issues were solved and you had to scale it, they had factories. There were 5 of them, made mainly in Boston, St. Louis, one in England, and Baylor in Texas. So they didn't have like 50 states involved because managing that could be
crazy.
Yeah, a little challenging. So they had 5 factories in the end that really did the production scale of the project. So similar sort of things. Whatever's most efficient to get the job done is the answer, but I know that's not a great answer. Yeah.
So currently, physicians can order pharmacogenomic testing to determine how a patient will respond to different medications, how they'll metabolize those medications. So can you speak to how the study of proteomics could add to this and how it could potentially help find the right treatment for the right patient?
Yeah, so pharmacogenomics is when you look at someone's liver enzymes, for example, that metabolize drugs. Because, Luca, you might metabolize the same drug differently than me. Mm-hmm. And I might generate a toxic derivative of that drug at higher levels. So the drug would act very differently for me. So that's the area of pharmacogenomics. And right now it's DNA driven. It's using the Genome Project. But doing the protein project would mean you would know all the different forms of those liver processing enzymes that metabolize drugs in the different cell types. So when I come to those liver cells and I know, you have elevated levels of these proteoforms, that is closer to the proteins that are actually doing the job, right? You're not using DNA and the potential of what's formed, you're actually using a test for how you are metabolizing drugs. So that proteins are closer to the trait of metabolizing drugs differently. So that's one example.
We have a question from the app, the C2ST2.cnf.io. The Human Genome Project sparked rapid advancement in technology in regard to DNA sequencing. What kinds of advances in technology do you expect to see in the area of proteomics?
Yeah, so the technological gains were tremendous in the Genome Project, and this had this very non-incremental mode where you just really take a leapfrog of an event. And so for proteins, this is the difference between bottom-up and top-down proteomics. So the Consortium for Top-Down Proteomics advocates for a more precise measurement of proteins. The field of proteomics is dominated by, maybe 85% of it is what's called bottom-up proteomics. And so you have this conversation in science, and this starts to really get at the real nitty-gritty of how we do these measurements. But right now, I have a metaphor for you, if you'll bear it, to answer the question. In stamp collecting, right? So if you're going to just collect 1 billion stamps, right? Or just from the blood. Let's say there's 20,000 proteoforms in the blood that you want to get and measure. Right now, you take those, and each one is a stamp. Right now, you take those stamps and you shred them into about 50 pieces each. And then you bring a big fan to the table and... Sorry, I didn't mean to scare you. But that fan blows pieces, and you only can collect between 5 and 10% of those pieces back. That is bottom-up proteomics. That's the dominant technology. Top-down proteomics takes each stamp whole, weighs or measures the whole stamp, and then breaks it down in a top-down strategy. That's the technology that the Consortium for Top-Down Proteomics is advocating for. That's the leap in technology we'd like to see. And this is the project that would bring that into being.
And again, I think I just want to reiterate again how important it is, again, for the public to be involved in this, right? Because again, it's the people that have to, regular common people have to understand how important it is. And then, like you said, the government can take that, run with it, and then along the way, the private sector gets involved, correct?
Public sector is critical for this. You asked about who would fund it, and the government is, it's the people, and to really ratchet up our understanding of ourselves at the molecular level, that's what this project's about. And the stakeholders are in this room, absolutely. And so I really appreciate all the questions.
So spread the word. Spread the word for sure.
Yeah, we got a few more questions if there's time, etc.
As a follow-up to the question about technology, when you're talking about proteomics and the different forms of the proteins that you can come across in the body, are you looking just at the structure of the proteins, or are you doing functional tests, or how are you determining what a form of a protein is? And in the process of getting it out to studying it, are you changing that structure in some way? And is that an advance that needs to be made before tackling the project, or do you think that's an advance that will come about out of necessity?
Wow, that's great. I think we have maybe a cell biologist in the audience, or a biochemist, and she threw out two great questions.
Sorry, I work on the PO1 with you. It's Daryl.
Oh, Daryl. Oh, hi. Oh, it's a plant. Wait, you're not. That's wonderful. Hi. Yeah, great. Thanks for coming out. So the proteins that you talk about, and what I'm talking about is not functional project. If you wanted to do a functional project, because proteins have enzymes, they have functions. What are they all? How much are they? The cost of that project is basically infinity. The cost of a function-based project. You also asked how to articulate a project. If you say, I want to know the functions of all the human proteins, well, you might as well just fund the National Institutes of Health for the next 100 years and we'll get there, right? No, this is a structural project. This is to ask, what is the composition of the estimated billion proteins that are come off of our genome in our bodies? I know, and it's really cool. It's really cool to talk about. It's the phone book. Let's make the phone book of the human body when it comes to proteins. So it's a structural catalog, and there's much more sophisticated ways to say what it is, but that's sort of plain spoken what it is. And how will that then lead us to all these functional insights and understanding human biology better? That's the leap that scientists don't generally make. But it is, when we come together as a society and we do projects like this, the moonshot, and I wasn't alive. Oh, no, it's true. I was alive. I was 1 year old in 1971.
There you go.
When we went to the moon. But the Hubble Space Telescope, we have learned incredible amounts about the cosmos through precision astronomy with Hubble Space Telescope. That was $10 billion plus the fix, right, of your taxpayer money. But we now know so much about the universe. And in cosmology or astronomy, those people get together and they play nice. They play for $5-10 billion pools of money. That's how astronomy rolls. In biology, really one of the few projects where that was true was the Genome project that's depicted on this slide. That was big science. And so will we come together for the human proteome? Is it functional enough? Are we able to make the leap of faith or leap of technology? Because the second part of your question was, do we alter the proteins that come out upon sampling? The answer is in top-down, far less do we alter those proteins than in bottom-up. Bottom-up, we shred them and we don't get all the pieces back. So that's the answer to your two questions.
We have one more question, Neil, and then we'll close things off.
Yeah, over the last 10 years, we've started to realize the importance of the microbiome in human health. Are you going to try and control for that or exclude it in some way?
Yeah, so like the Genome project, we'd focused on the genes from the human, Homo sapiens, our genes, and domesticating, regularizing that. So the answer is no, not the first iteration of the project. The microbiome has been close to a big science project. The microbiome has been very well socialized. There's a lot more cells of bacteria in your body than your own cells. But I would assert that domesticating our cells and just what is encoded from our genes is important enough to focus on in isolation. But the technology that we develop can immediately be applied to bacterial proteins. And the last thing I'll say is that, Luca, I love you, man. Thanks for coming out.
No problem, man. No problem.
That's the last thing.
Anytime.
Right. You don't like play Neil Diamond, short people got no reason. You could.
I could, yeah.
But you don't.
Yeah.
I appreciate that.
I thought about that song, Wish I Was a Little Bit Taller, I Wish I Was a Baller. You know that song? I asked Chris to put that as your walkout song.
Okay, we're going to fade
song.
to that song, apparently.
Okay, all right.
I love you guys.
Okay.
That's great.
Okay.
But bacteria, they have maybe 5,000 genes, down to 2,000 genes. And when they create a protein, it's basically very much corresponding to the gene. That is not true of human beings. Human beings, we have the same number of genes as like a worm, a little worm in the microscope, or a carrot. We do not have that many genes. What's complicated in us is that from the same gene, we can create all these different proteins, because one proteins will decorate and change the others. That's partly what we're trying to unlock in the Proteome project. So I really want to thank C2ST and everybody for coming out. If anybody happens to have a couple $1 billion, throw it in the basket on the way out.
We take Venmo, Quick Pay for all those. Yeah.
Right. Yeah.
We do have one programming note. There's an evaluation for today's program that you can fill out at c2st2.cnf.io. We ask you to join us for that. And also, Neil and Luca will be available after this program for your continued discussion. We'll make sure they have a beer in hand so that you can continue this discussion.
Yes.
Thank you for joining us. You've been a great audience.
Thank you, guys.
Good night.